In this lab, my group investigated if plants in the sunlight will have a higher stomatal density compared to those in the shade. Stomata are found in plants to help the plant regulate dehydration. In a pair of guard cells, there is a hole created between them which is called a stoma. Multiple stoma are referred to as stomata. These guard cells can either close or open the mouth, and if the mouth is open, there is continuity between the air and atmosphere. Therefore, the stomata being open makes it possible to lose water through the open stomata. The open stomata are necessary to generate a negative pressure that will help pull water up through the xylem of the plant. The xylem is a tissue in the plant that moves water up through the plant. As you can see in our graph, there were 18 open stomata, pictured on the left, on the plant in the sun, and 14 open stomata, pictured on the right, on the plant in the shade. Based on our title, the number of open stomata based on setting, we looked at whether the Zebrina plant was being placed in the sun or the shade. The conditions in this lab were constant, with the only variable being the sunlight. Sunlight is the independent variable because it is the changing factor that is related to our hypothesis. As shown in the graph, the plant in the sun had more open stomata than the plant in the shade. After testing the stomata in both the sunlight and limited sunlight, it is apparent that the stomata are more likely to be open in more sunlight as opposed to limited sunlight. This would coincide with our hypothesis because we predicted that plants would have a higher stomatal density in more light as opposed to those in the shade. The result is likely due to photosynthesis. In the shaded areas, the plants can't go through photosynthesis as easily due to the lack of sunlight. Therefore, in order to prevent the plants from becoming dehydrated, the plants will close their stomata to save water. As a result, the overall stomatal density will be lower due to the lack in sunlight.